Okay, welcome back. We are going to finish up, I promise this time, on the chapter 11, and this will be part 3, and looking at long-term infections and long-term effects. There's some terminology associated with these sorts of phenomenon. Latency is a dormant state of microbes in certain chronic infections. You may have heard or experienced or whatever. You may get a particular infection, and if stress or something later on down the road happens, that all of a sudden you'll get a reoccurrence. And sometimes this will bring it out. Herpes is a great example. Simplex, herpes zoster, Epstein-Barr, those sorts of things. And syphilis, typhoid fever, malaria, and others also experience a latency. So viruses and other microbes like bacteria and protozoa all follow kind of similar things, where they can kind of step down in terms of their infection and kind of sit dormant for a while. Sequelae is the long-term or permanent damage to tissues or organs caused by infectious disease. Meningitis ultimately will lead to deafness, and strep throat can cause a rheumatic heart disease, and we're also seeing sort of a bilateral numbing effect in children with the hands and feet. Sometimes they get resolved, and sometimes we're looking at not. Lyme disease with arthritis, Bell's palsy, which the nerves actually swell, although the bone openings, the where nerves pass through the foramens and the like, they swell and then they lose innervation, and then the face goes numb, and then ultimately that comes back. But Lyme arthritis is a permanent type damage. Polio, or we lose some of the nerve function and paralysis due to that infection. Incubation period. This is the time from initial contact with the infectious agent to the appearance of symptoms. And again, we're using this example of the COVID-19, where we see the initial contact and we have to wait 2 weeks to see if we see coughing, loss of smell, of being able to determine various smells and things, some of the strange things associated with that virus. Agent is multiplying at the portal of entry but has not caused enough damage to have the symptoms, if at all, depending if you're going to be asymptomatic. Varies according to the host resistance, the degree of virulence, the distance between the target organ and the portal of entry. A lot of these have a direct impact on the incubation period. It can be several hours to several years, depending on the infectious agent. Lyme disease, we could have Lyme, and we're looking 6 to 8 months down the road before you start to see some of the symptoms. A majority of infectious range from 2 to 30 days, usually are typical. Palindrome, running forward. The word means prodromal, not palindrome. Prodromal, sorry about that. Running forward a stage, a 1 to 2 day period where the earliest notable symptoms of infection appear. And everyone's experienced this. If you get, let's say, some sort of flu or something, something's not right. You start to feel, weak or tired, or your eyes squeak when you rub them. I know that sounds weird, but some claim that that's sort of the prodromal type of experience. Muscle aches, fatigue, upset stomach, general malaise, or feeling kind of draggy. The acute phase, the infectious agent multiplies at high levels and exhibits the greatest virulence and becomes well established, marked by fever and other prominent types of signs and symptoms. Extremely variable in length and period. So that's pretty straightforward stuff. The convalescent period, where the patient begins to respond to the infection and the symptoms decline, usually like viruses where they're self-limiting. Patient strength and health gradually return due to the healing. Many patients stop taking antibiotics, unfortunately, during this period. If you're dealing with bacterial infection, though the pathogens are still in their system, leading to an antibiotic resistance. And in other words, you didn't get rid of them, and the antibiotic dosage goes away in the blood, and now the antibiotic is gone technically, and then the bacteria start, the ones that survived it are now much more resistant to it next time, because the ones that survived are the ones that had that borderline resistance, and it just improves over time. So either the organism lingers for months or years or indefinitely in the continuation phase, or the organism's gone but symptoms continue due to some lasting effect. So here's the stages of a disease. Here's the incubation period, initial exposure here. We have that prodromal stage. Well, I feel a little lethargic, something's going on. And then you get the acute phase where the multiplication, whatever the agent is, increase, and finally the immune system starts to deal with it. Your body will start dealing with it, and then you'll go through this convalescent period, and either you get rid of it totally, or based on some, if it's gone intercellular or it's gone to the joints or something, you'll have a continuation period depending on how it was treated. Reservoir, a primary habitat in the natural world for which a pathogen originates. I've always been curious about various diseases and where their origins are, and Ebola disease comes to mind, and those sorts of things. Where do these diseases come from? One of the theories I've heard was because of the growth and the establishment of building homes and various things, we move out indigenous populations of living things like bats and other animals that live in those, and there's sort of like a little microcosm of disease agents that they pass from one another, and then you break down the integrity of that little community, and you may now be exposed to a particular bacterial or some kind of pathogen, viral, that hasn't really ventured much outside of that community. It is somewhat virulent to those animals, and it could be zoonotic. In other words, it can travel from human to animal or animal to human. And then we start to see these diseases that are rather extreme, and ones we haven't seen before, because of these small communities. Now, one could argue that that's could be part of their defense mechanism, believe it or not. I mean, there are examples of where different organisms kind of protect each other. Fire ants on a particular tree, and a giraffe will start to eat on the vegetation, shaking the tree, and then the fire ants will go up the snoot of the animal and have extreme discomfort because the giraffe is experiencing these fire ants biting them, and the giraffe will stop eating the leaves. Now, in service for that, in-kind treatment, I guess the plant will give sugars to the ants. So things like this can occur, and although there may not be explicit examples of this in the bacterial world, I could bring your attention to the probiotic, where there's synergistic positive effects of having some microorganisms that do some protective value. So it's not unheard of, and it's not that far-fetched. Transmitter, an individual or object from which an infection is acquired. Syphilis, well, that's kind of obvious. And having someone that has syphilis and then continues on with certain activities will now become a reservoir and a transmitter. Hepatitis A And the reservoir is a human, and then of course, if you don't wash your hands after using the bathroom and that sort of thing, then you transmit it through the food, which is not a very pleasant concept. So reservoirs and transmitters are really, really important to try to understand and follow those. Pathogens from animals can be directly transmitted to humans, as an example of bats transmitting rabies to humans. It can be transmitted to humans via vectors such as fleas passing the plague from rats to people, well documented. Can be transmitted through vehicles such as water, such in the case of leptospirosis, which is uh, a really painful and life-threatening disease, in which often transmitted from animal urine to human skin via bodies of water. Uh, a person suffering from a cold contaminates a pen, which is then picked up by a healthy person, and then that person starts chewing on the end of a pen or a pencil and then transmits a disease. I don't think that happens. I've seen students do it in the microbiology lab. I get a little upset when I see that. A person who's fully recovered from hepatitis but is still shedding hepatitis in his feces may use suboptimal, or if any, hand washing technique. Now, the general rule, and this has been studied by several institutions, where about 60% it's been observed of men that finish with their whatever in the bathroom don't wash their hands. Now, it's sort of inverse in women. It's about 40% don't. So it's not a very good contemplation, but anyhow, it is what it is. Carriers can also transmit through direct means when incubating carrier of HIV who does not know he or she is infected transmit the viruses through sexual contact. That's not the only one. When an arthropod is in the host and reservoir of the pathogens, it's also the mode of transmission with arthropods and vectors that we know of. Some pathogens, such as tuberculosis, can survive long periods in non-living reservoirs and then come directly transmitted to humans when they come in contact with contaminated soil, water, or air. Another one comes to mind. It's Legionnaire's disease. It's usually in ductworks and cooler air conditioners and things like that. And then because of the location, it's spread by aerosol via fans, and this organism now will infect and potentially cause death. A great number of infections that affect humans have reservoirs other than humans. Persons or animals with symptomatic infections are obviously sources. So a carrier is an individual who inconspicuously shelters a pathogen, like the COVID-19 virus, spreads it to others without any notice, and may not have experienced the disease due to the microbe, whatever reason they're not infectious. So here are the carrier states: asymptomatic, incubating, convalescent, chronic, and passive. And just to go through, asymptomatic is the infected but show no symptoms. Gonorrhea, genital herpes with no lesions, human papillomavirus. Incubating carriers spread the infectious agent during incubation period, like infectious mononucleosis. Convalescent carriers are recuperating patients without symptoms. They continue to shed, like hepatitis A and having bad bathroom habits. Chronic carriers, individuals who shelter the infectious agent for a long period after recovery because of the latency, like tuberculosis and typhoid fever. And in passive carriers, medical and dental personnel who constantly handle patient materials are heavily contaminated with patient secretions and blood risk, picking up pathogens mechanically and accidentally and then transfer them to other patients. And this is, where we use catheters or other things more than once, that without sterilization or anything, and pass certain things on to others, and which is 100% avoidable, but that takes some careful considerations there. obvious ones. The majority of animal reservoir agents are arthropods like fleas, mosquitoes, flies, and ticks. Larger animals can also spread the infection, mammals and rabies, bats, birds, parrots, psittacosis. Uh, Lizards, salmonellosis could be those little turtles, pet turtles, and kids kiss them and then get salmonella. Biological vectors actively participates in the pathogen's life cycle, serving as a site for which it can multiply and complete. So a biological vector is one that contributes into the life cycle of whatever the pathogen. We see this with schistosomas and other types of diseases and things, which we'll talk about. Mechanical vectors carries the microbe more or less accidentally on body parts. It's just there. It's contaminated. It doesn't bother it, but passes it around. And then I've already mentioned the word, but zoonotic or zoonosis is an infectious agent of some kind that's indigenous to a particular animal, and it can naturally transmit the disease to humans and back and forth. Humans are essentially dead-end hosts that do not contribute to the natural persistence of a microbe. Some zoonotic infections have multi-host involvement. Some have very complex life cycles. Zoonotic spread of disease is promoted by close association between humans and animals. One of the studies I was involved in is I studied diarrheal disease, and we were looking at the farms. Were farmers getting diarrheal disease from handling the farm animals, and then were the farm animals susceptible to the farmers perhaps in passing along? And we found out that they were not in the particular organism E. coli that I studied with the heat label or heat stable toxins that harbored those organisms of E. coli did not transmit, and we did that genetically, and we could show the signatures were different between the human and animal sickness, so they were isolated. So sometimes it does, sometimes it doesn't, and you have to kind of follow up and see what's going on. Make up about 70% of all new emerging diseases worldwide are zoonotic. That's kind of a disturbing concept. I don't know why that is. I could hypothesize, but that doesn't do anyone any good. Impossible to eradicate with also eradicating the animal reservoir, which has happened when we see a particular disease, a really nasty one in horses, and trying to deal with it, they just destroy all the horses. The equine encephalitis is a good example where they'll just kill the entire farm worth of horses to prevent it from ever spreading, because it's such a horrible, it's an Ebola-like disease in horses, and it's really nasty. It spreads really fast. Unfortunately, you have to get rid of the animal in cases like that. Common zoonotic infections: rabies, yellow fever, viral fevers, hantavirus or rodents. And there's several examples of that even in the U.S. As we speak. Influenza, chickens, birds, swine, chickens, birds, and pigs. West Nile virus, we're seeing more and more of this, wild birds and mosquitoes, and causing problems. Rocky Mountain spotted fever with dogs and ticks, a different kind of tick from the Lyme disease tick, but rickettsial disease caused by rickettsii, psittacosis with birds, leptospirosis with domestic animals, anthrax, domestic animals like sheep and that sort of thing, brucellosis, cattle, sheep, pigs, the plague spread by rodents and fleas, and we actually are seeing cases of this in California with some of the situations of folks living in the streets there. Pretty scary. Salmonellosis, mammals, birds, reptiles, and rodents. Tularemia, which is sort of a worm type of infection. We'll talk about rodents, birds, and arthropods will spring. Now, ringworm, which is a fungal infection, domestic animals and humans. Toxoplasmosis, cats, rodents, and birds. We see sometimes when you don't wash your hands after changing cat litter boxes and things, humans can get toxoplasmosis, trypanosomiasis, depending on how you emphasize some of the words. Uh, Domestic and wild mammals, trichinosis, and this is in swine, pigs, and bears, and it's a parasite that goes into the muscle tissue. It's one that was written up in the Bible with eating pig or swine food that as a food. Tapeworm, cattle, swine, and fish. And tapeworms, is of course in dogs too. We see that. Microorganisms have adapted to nearly every habitat in the biosphere. We thrive in soil, water, and air. Surfaces in homes, offices, and other structures in the built environment. Most are saprophytic and cause little harm and considerable benefit. Some are opportunists. A few are regular pathogens. Because humans are in regular contact with environmental sources, acquisition of pathogens from non-living reservoirs is always possible, and this is something you have to be careful of when you go camping and things, when you're out of the environment, or go swimming in water holes that you think are clean and may get a transient infection, or even worse with flesh-eating bacteria. Communicable is a disease in which an infected host can transmit the infectious agents to others. And this is incommunicable diseases of what we're living through right now. And it's infectious and is sometimes interchangeable with communicable. So infectious usually is associated with bacterial. We know that viruses are communicable, most of them, but this is not a precise usage, and it's because of it's been around for a long time. Contagious is a disease that's highly communicable even through direct contact. So, wearing face masks and gloves and various things, I think everyone knows that drill. Influenza and measles are highly contagious, as would be COVID 19. Hansen's disease or leprosy is only weakly communicable because of the way it's transmitted. Non-communicable are infectious diseases that does not arise through transmission of the agent or the of the host, and the individual has accidental contact with a microbe that exists in a non-living reservoir. Infected persons do not become a source of disease to others, and they're non-communicable. That's pretty obvious. Patterns of transmission, direct or indirect, with animate or inanimate objects. Horizontal transmission is a disease spread through a population from one infected individual to another, and that's what we're living through. Three major modes: direct contact, indirect contact, an object or substance carries the agent. That's why we wipe down things like mail and things like that. You touch it. And vector transmission, arthropods that harbor an infectious agent transfer it to human. Vertical transmission is disease transmitted from parent to offspring via ovum, sperm, placenta, and milk. And there are several diseases that are transmitted in such ways. So vertical transmissions from parent to offspring, ovum. Horizontal is disease spread through population, as we just said. Healthcare association infections, or HAIs, are not as common infections. Infectious diseases acquired or developed during a hospital or healthcare facility stay. From 1% to 20% of all admitted patients, with an average of 4%. And the healthcare associations are really trying to dampen these down by making sure rooms are cleaned with UV light sources to zap bacteria and that sort of thing. 750,000 cases a year, resulting in 75,000 deaths, and that's unacceptable. There can be further work done on that. Factors tied to healthcare associated are compromised patients. I mean, obviously, you're servicing a realm of people who are not well. Collecting point for pathogens, of course, that's why people go there aren't well, they're sick. Lower defenses permit normal biota to enter the body, which we know. Infections acquired directly or indirectly from fomites, things contaminated, medical equipment, other patients, other medical personnel. One of the studies I remember, it just stuck out in my mind for this, is one of the sources of infection from patient to patient turned out, oddly enough, to be the doctor's tie. He'd lean over in a tie by gravity would touch, and they were asked if they ever washed their ties or cleansed them and stuff, and the answer was no for about 90% of the time. And wow, really? So that's been dealt with in some cases. Visitors, air, and water. It makes it quite interesting. Healthcare processes that lead to healthcare associated treatments using reusable instruments such as respirators and thermometers. My God, please make sure those things were clean or disposable. Indwelling devices such as catheters, prosthetic heart valves, grafts, drainage tubes, tracheostomies, would stick a tube. They are usually considered invasive, and they go into, of course, a portal of entry, and they can be contaminated, and sometimes are. A lot of the prosthetics, like heart valves or hip replacements, contain a lot of areas. If they weren't thoroughly cleaned, autoclaved, and that sort of thing, there's lots of nooks and crannies for microorganisms to hide out and survive, perhaps some of that process. High proportion of the hospital population receives antimicrobial therapy, so drug resistance are usually much higher in the hospital setting. Most common types: Clostridium difficile. That generates a code brown that will spray a wall. Staph aureus, pneumonia, surgical site infections, multiple resistant Staph aureus or MRSA, wounds that don't seem to resolve and get worse. Klebsiella, surgical site, urinary tract infections, pneumonia. This Klebsiella will produce a lot of polysaccharide and cloaks itself from the immune system and hides from the immune system because it covers itself with sugars. And Klebsiella is really famous for that. E. coli causes urinary tract, surgical site, and bloodstreams. It's considered one that can grow invasively, given an opportunity and a little bit of help getting in internally. It'll do just fine. Enterococcus species, usually surgical site, urinary tract, bloodstream. Enterococcus has been implicated in transferring high antibiotic resistance to other microorganisms. There's association, they're figuring it out. It seems that they're able to do that and persist themselves. Bacteria will share a lot of genes and do it without any, they don't care, they're not shy. Most common healthcare: pneumonia is 22%, surgical site 22%, gastro or GI illness about 17%, UTIs 13%, blood infections, which always takes a more aggressive type of organism, about 10%, and then ones that they can't really determine around 16%. So pneumonia is up there along with from resulting from operations and that sort of thing. Of course, the controls are done by infectious disease officers or infection control officers, depends on what people name them. Proper practices and procedures, SOPs, are followed. If not, then people are let go. They are trying to fight these diseases, track potential outbreaks. There is reporting requirements, and you got to follow those. Special training needs to be given that you'll get, like how to put on PPEs and that sort of thing. All sorts of different types of training that you might get depending on where you're working, where you're regular exposed to needle sticks, infectious secretions, blood, physical contact. And a lot of the hospitals have these SOPs as universal precautions that they may or may not see it, but it doesn't matter, they're going to practice it anyhow. And that way, in case you get blindsided, you're already practicing. So which agent is the cause? That's always the question, and I've been involved in a lot of cases where we're trying to find out what the causative agent is, and it one of the things you use is Koch's postulates, and it came early on when we're trying to study infectious disease and understand what's going on. Um, it does play a continual role. Some virologists are trying to apply it, which Koch's postulates doesn't really apply to viruses because you can't culture it. One of the steps is talking about in Koch's postulates culturing the microorganism, and it's really hard. You could do it in cellular cultures, but they're not doing it on their own, so it technically it's not along the lines of Koch's postulates. You can sort of modify it a little bit uh, if you want to take that read on it. Reliable for many diseases, but cannot be completely fulfilled in certain circumstances like viruses. So Koch's postulates: you isolate the microbe, infectious subject, and cultivate it. Get it in pure culture. Characterize it best you can, and then you inoculate it into a susceptible host and observe the same sort of the resultant disease. Once the organism is recovered, you reisolate, and you see that it's the same organism. Then you know it's followed the Koch's postulates. The problem is that some infectious agents can't be readily isolated or grown in a laboratory. That's the case for some STDs, a sexually transmitted disease, like syphilis. I was in an environment where the only way to grow it was to grow it in the testes of armadillos, and it's not like we got plenty of those for growing it. And when I was working at Burris Wellcome, what they wanted to do was to grow it in a medium so that we wouldn't have to resort to that sort of thing. And that took about 2 years of research, and finally we did find media to grow it in, but it's hard in some cases. Viruses have a limited host range. Human viruses will only cause disease in humans, and etc. And you have to grow it in a cell line, which is not the same. So it's very hard to cause that. And if it's a polymicrobial disease, and another good example would be Lyme disease, because there's about 8 different organisms that can contribute to Lyme disease, and the Borrelia burgdorferi is best studied, but that's not the only organism, and sometimes they think that having other organisms present adds to the pathogenicity of the overall Lyme disease. So we're going to look at epidemiology, some of which you probably picked up with the recent epidemic. Explain what is meant by disease being notifiable and reportable. Define incidence and prevalence and what those terms mean. And 3 major types of epidemics, and identify the epidemic curve associated with each. So we'll do that best we can. So the epidemiology is the study of frequency and distribution of disease and other health-related factors in defined populations. It involves many disciplines: microbiology, anatomy and physiology, immunology, medicine, psychology, sociology, ecology, statistics, genetics. I think that's what else. There's a few that weren't really mentioned. Well, the study of microbiomes and things like that are all disciplines. Consider all forms of disease: heart disease, cancer, drug addiction, mental illness. Various long-term and short-term types of diseases can all be caused by various contributions and interactions. Florence Nightingale laid the foundations for modern epidemiology. Now let's talk about that just for a second. Florence was she was just amazing note taker. She wrote details about everything, spent the time, and so she understood that filth contributed to disease, and she wrote down every detail. And because of that, Florence Nightingale was able to follow a relationship of soiled and dirty things to disease. And that was sort of, I mean, think about it. In the day, people would be walking down the street, and somebody upstairs, the second, third floor, would just empty their little tray. They used to use the bathroom in a little things, trays, and then they chuck it out the window, and whoever's walking below, well, that's too bad. And so you can see that disease might have been communicated a little bit easier in those types of environments. I wouldn't want that today. So uh, very meticulous notes is what I'm saying about Florence Nightingale, and demonstrated that more men died of disease than traumatic injuries. So diseases are something to really to identify with. Uh, Techniques of the epidemiologist is to identify causative agents using adaptations of Koch's postulates, track behaviors, collect clues of causative agent pathology, track the numbers of distribution of cases, which they've done with COVID. Who was the patient zero and that sort of thing, and then who did they come in contact with? All this in a huge expense to do that, and it's done for tuberculosis. It's done for a lot of diseases, and it all comes to contributing to the public health departments and finding out what's important. This is a biggie: reportable and notifiable diseases. There are certain diseases by law that have to be reported, and the idea is that we want to prevent the spread and having a epidemic like we're experiencing now. Other diseases are reported on a voluntary basis, but usually hospitals are really good about reporting. Network of individuals and agencies at the local, district, state, national, international level keep tracks of these infectious diseases. So I'll give you a few minutes to memorize these. No. What we do is that we follow lists of these, and they're usually automated and part of the system. When you report that a patient has a particular thing, the reporting is usually done automatically. There are some diseases here you may not know, and I'm working on a case study right now. Powassan disease, we're going to look at that being spread by ticks. Isn't that nice? There's several other diseases spread by ticks that can occur all together. And so the papers or the case study is called a ticking time bomb, playing on the fact of tick and ticking time bomb. And the idea is that we have, I'm going to talk about three different Morgellons disease is spread by ticks, and talk about that as a very bizarre disease. Let's see, and of course, Lyme disease, and those are some serious diseases, and we'll talk about that in that review. Just going through the list, you can see all sorts of diseases you may or may not be aware of. We're going to talk about some of these, the body systems that are affected by these, and that sort of thing. So here is some statistics that we need to know about as reported by the CDC and other agencies. And I'm going to give you a report that you can follow. I don't know if I'll add it as an extra credit or not. I just want you to be aware of these free resources where you can obtain statistical information if you want to know for yourself. You constantly want to be notified from the CDC what might be coming out as prevalent, and get on morbidity mortality reports. I know that sounds bad, but it's really important to know what's coming through the door so you can be prepared. And so the CDC has a lot of resources that you just need to know how to get to them, and I have some worksheets that will show you how to do that, and you can just keep them for future reference. We just don't have enough time this summer to go over that. Total number of cases in a population divided by the total number of persons in a population is the prevalence. And of course, we've been running through all these on a daily basis about COVID-19 ad nauseum, but it's important. Incidence is the number of new cases over a certain period of time. And the incidence rate, of course, with COVID-19 we want to know about, because that determines if we can open up various things or not. We don't want new cases, we want to decrease in cases. Statistics of concern to the epidemiologists, rates of disease with regard to sex, race, or geographical region. Mortality rate measures the total number of deaths in a population due to a certain disease, and overall death rate from infectious disease has dropped, although the number of persons affected with infectious rates, mortality rate, has remained high. And all of these terms you should be aware of just by watching the news, if you're so willing to do so, and you get this on a daily dose sort of thing. So the depictions of the data, it's usually done graphically, and you have usually along the bottom of the x-axis we have the year, and the y-axis we have reported cases or incident rate or various things, or you can have a plot based on some sort of range of color, and you'll see a graphic such as this looking at the Zika viruses in 2017. And obviously, if you're a hospital in New York, this is a concern you want to have when children are born, to see if there's defects and things, or screening for it in pregnant women to see if there's a way to counteract if there is going to be an incidence rate of that particular disease. Epidemics, we have common source epidemic results from a common exposure to a single source of infection over a period of time. Propagated epidemic results from an infectious agent that's communicable from person to person and sustained over a period of time. Point source epidemics is infectious agent came from a single source and all of its victims were infected at once. So of course what we're living through right now is sort of a hybrid of these. We have a common source that was propagated throughout the world, and I've never seen anything quite like that. Epidemic curves, you can see in 2013 there was a outcropping of particular person's cases that were rapid onset. It doesn't say what the cases were, but it doesn't matter, it's just for illustrated. Positive specimens we saw perhaps 2015. You see the ranges, and they just use these types of plots to show trends and that sort of thing. So numbers of persons versus year, x-axis and y-axis, you can see, and usually that's a standard way they represent these. Additional terms: index case may not be the first case of the disease, but it's the first case that brought epidemic to the attention of officials. Usually they want to see who the person that started to spread it or something like that. Endemic is an infectious disease that exhibits a relatively steady frequency over a long period of time in a particular geographical locale, and having perhaps Ebola disease in an African area, that could be an endemic type. Sporadic is occasional cases are reported at irregular intervals at random locales, and it could be Zika virus or something like that. Epidemic, where statistics indicate prevalence of an endemic or sporadic disease increasing beyond what's expected. Or pandemic, which I won't go and give an example. I think it's excruciatingly obvious. Spread of an epidemic across continents. Very rare. So that's it. That's all that we have for Chapter 11. I wanted to make sure that Chapter 11 was one of those that gave you a broad exposure to, well, that's just a term, to the understanding of why we need to study some of the details that we are and how it contributes to disease. My students always like to know why we study certain things, and so that's why we jettisoned out to Chapter 11 without we got some important terms that we're going to learn that we have learned from these chapters. And what's really nice about that, though, is it kind of gets you ready and preps you for the discussions and more details of some of these things. So I think logically Chapter 11 was a good one to go through. It wasn't that difficult in concepts or terminology, but are words that we are going to experience again and again throughout the course. And again, unfortunately, we do a lot in a week, 2 weeks worth of work. I'm trying to get us up to speed without ramping and just kind of mindlessly going through it. I want you to systematically go through it. So that's where we are. We'll be picking up on chapter 2 and 15 this week. They're not as in depth as long as the chapters, and I knew that, and that's why I spent a little bit more time on 11 to give you that basis. And as we get through 2 and 15, it just keeps hammering away the concepts of things that we need to know. Now, chapter 2 is focused on the lab, and that's I kind of dragged my feet a little bit because the kits haven't come for the majority of students. And so what I'm trying to do is we'll start the introduction for the lab with chapter 2 that brings up a lot of the concepts of the lab, and then I will have presentations that I'll turn on that go over what's in the lab manual in much more detail and give you that lab experience. And so I wanted to get a little bit of vocabulary and get you comfortable going and reading and doing things. It will be the same sort of philosophy. The only difference between the lab and the lecture part is the lab has lab practicals, and we have two, and that's where about 25%, 30%, I can't remember off the top of my head, of your grade is from the lab practicals and the lab part of the course. And so that's why I'm trying to be really careful as to when I spend time on the lab, it's good quality time. And so we've got some topics to go through, safety and those sorts of things, which are important. Some of the general characteristics of the lab that we're going to have this summer. And then I'm going to go over the microscope in a lot of detail, and hopefully you'll have a good experience. I have my own microscope at home, and I'm going to tear into it and show you the pieces and parts, and you'll be able to see exactly what I see on the screen because it has a camera out on it, and you'll be able to see what I'm doing, focusing and the like. So it's the best next thing to being there. It's the best I can do, but I hope someday that you'll get an actual experience. I have some virtual microscope work that you can do. Most students feel that it's just not the same, which I know. We're just trying to get by. They're doing the best, but I think I can illustrate at least the things that you need to look for. So we've got this week to do. It should be fun, and we'll go from there. Again, I appreciate your time and all your hard work. You're doing fine. Let's keep it going, and I'll get back to you now with chapter 2 this week.